Welcome back, ladies and gentlemen. Today we're going to spend a little bit of time talking about these mitotic spindles or these spindle fibers because they really are a hallmark on how this whole process works. We need these things in place. And if they're not there or not doing their job, then the entire ability for your cells to separate that genetic information pretty much becomes non-existent. And in order to do accurate cell division, we have to be able to get those two daughter cells to get the right amount of genetic information into each new cell. So when we talk about mitotic spindles, right, these are these microtubules, so they're part of your cytoskeletal structure that really do control that chromosome movement during mitosis, and to be fair, they'll also show up during meiosis as well. Um, and these microtubules, they arise or are built from these centrosomes on either end of the cells, which is where those centrioles are located. Those are your microtubule organizing centers. So these are protein fibers, right, that are being constructed in your cells specifically to move those chromosomes during cell division. So when those centrosomes, which include the microtubules, include the centrioles, move to this poles, right, we go through cell division. Each new cell will get one set of these, so when we go prepping for cell division during G2 in interphase, we've got to duplicate them, and then throughout prophase, they end up migrating to either end so that we can accurately then create those microtubules, attach them to the kinetochores here that are on those duplicated chromosomes so that we can accurately then pull these guys apart once everything locks in place on that metaphase plate. So if we look at these sort of centrosomes, these organizing centers, this is kind of what you end up seeing when we look at these guys. So you've got your centrioles, which will sit here in the middle. You've got your microtubules that will then extend out from there to the chromosomes that will line up in the center of your cell, what we know is your metaphase plate. And then you also have on the back end of those centrioles are these asters, or this array of shorter microtubules that are going to extend from each of those centrioles, and they are going to help to also sort of anchor everything in place. So those spindle fibers will go towards those chromosomes and attach to those kinetochores, right? And that's going to happen during prometaphase. And then once all of that is attached, everything snaps into place during the midway point of the cell, that metaphase plate, during metaphase itself, so that everything then should be equally divided across that plate as we start to move those chromosomes towards those centrosomes. So here's an interesting thing. I've been mentioning this term kinetochore, right? This specialized region that's attached to the centromere that links your sister chromatids together, right? These are kind of, I say kind of think of them almost like little hooks or little Velcro components that the microtubules then attach to. You also have these non-kinetochore microtubules, all right? And those are back here um, also working as part of that aster, as well as also being stretched here non-overlappingly in the cell itself. And those non-kinetochore microtubules are going to help to push the opposite poles and help to elongate the cell so that when we are ready to have all of this material separated, we can then more easily go through, use some additional microtubules to then split the cell if we're talking about an animal cell, or create a region that is capable for the cell plate to form to separate the plant cells. So the actual kinetochores themselves are the regions where we're attaching those centromeres. We get these non-overlapping um, non-kinetochore microtubules then that help to push the cell, stretch the cell, so that we have enough space to actually then divide in the center when it's time to go through cytokinesis. Okay, so here's an interesting question. how do these spindle fibers and the connection to those kinetochores and the connection back to the centrioles, how does this all actually move those chromosomes? Because that is really their ultimate job, to separate out the genetic information and get that information, get those chromosomes to either end of the pole so that we do go through cytokinesis. Each new cell that is created as a result of mitosis has enough genetic information to function. Well, we had to figure that out. All right. And this is actually a relatively recent discovery within the last 15, 20 years. The original thought was that these centrioles sort of acted like a winch on a car, right? Where we'd have rope tied around it or cable tied around it, and you'd reel in whatever you were pulling, or like the reel on a fishing rod. And so that was the initial thought, is that we just kind of yanked these guys in. This is where those microtubules were being formed from, and as a result, were being broken down from as well. But what we found out, which is what this diagram here is showing us, is that it's actually the opposite. That kinetochore almost acts kind of like a Pac-Man, and actually eats or degrades, breaks down the microtubule protein, so it depolymerizes that microtubule into these tubulin subunits as it's moving. So we've got motor proteins here in part of that kinetochore. It's breaking down those tubulin subunits as it moves across it, and they actually move from the center out. They break down that microtubule as they go. And the reason that we managed to figure this out is that they did experimentation where they actually created breaks in those microtubules in the cells. Presumably that would have assumed that this component would have kept moving and the chromosomes would have arrested where they were. They would have stopped in their movement. And yet we didn't see that. They kept moving, and these microtubules back here actually didn't do anything. And so in anaphase, as those chromatids split, as we break apart the proteins that are holding those sister chromatids together, that kinetochore then actually eats away those microtubules using those motor proteins, walks that chromosome down that microtubule as it's being depolymerized on the way out.